in the south of Spain, in Malaga, where our institute is located. And we have two different institutes, one in the city of Malaga, another one in Algarrobo Costa, which is another 50 kilometers away, where we have our experimental station with germplasm collections of subtropical fruit crops. And these subtropical fruit crops, which are the crops we work with, are mainly three: avocado, mango, and cherimoya. And so if we talk about their importance in terms of cultivated hectares, avocados are by far the most cultivated ones, around 15,000 hectares cultivated, and they consume around 6,000 cubic meters of water per hectare and year, producing 8 to 16 tons per hectare. And the second rank is mango, with more or less 5,000 hectares cultivated, and they actually consume 4,000 cubic meters of water per hectare in year, and they produce around 14 to 25 tons per hectare. And the cherimoya is actually a crop which is less important in terms of cultivation, but Spain is still the most important producer in terms of agronomy and also exporter, although most of the market is local market. And it's around 3,000 hectares, and they consume 3,500 cubic meters of water per hectare and years, and producing 12 to 15 tons. Okay, so the questions we address here are have two angles. One of them that I like a lot is evolutionary, and the other is agroecological. Evolutionary from the perspective that we work a lot with species that potentially have the ability to tolerate or resist the stresses. For example, polyploids. Over the evolutionary history of plants, polyploidization was actually one of the events that lead to more resistant varieties or more resistant species. And actually, polyploidization, which is basically the multiplication of chromosomes in an individual, either by failure during the gamete formation, leading to diploid gametes, and then their fusion will produce a polyploid, or by allotetraploidization, which is basically made by hybridization. And so polyploids have been used in agriculture for so many, for centuries, I would say, or decades, maybe. For example, strawberry is a typical polyploid, a typical octoploid. And so the effects of ploidy in the morphology of organisms are increasing fruit size, but also increasing organ size. And but this is not always linear, because the increasing organ size associates with ploidy sometimes. So that means we need to study species by species, although cell size is normally related to ploidy. And we are interested in the cell size of the vascular system, because the vascular system is where most of the water is running through in the crops we're interested in the fruit crops. So if we look at an ideal tree, for example, and roughly the 70% of water is actually transpired, but the 30%, the remaining 30% is actually used to transport photoassimilates through the phloem. And this is the tissue we are mostly interested in. And this is the tissue I'm going to talk about today. The phloem, just a reminder, is a tissue composed of individual cells that connect in series. And so basically, they operate like an electric circuit. In the source organs, the photoassimilates will enter the seed tube elements, increasing the osmotic pressure, and that makes water to get in the cells. And at the same time, in the sink tissues, for example, in the roots, the photoassimilates will exit, dropping the osmotic pressure, and therefore the water will go out. That creates a flux, and this flux has a velocity. And the explanation, the simplest explanation for this velocity is the Hagen-Poisseville equation, which states that the velocity is depending on several factors, but the ones we are interested in are the conductivity, or in other words, the geometry of the individual elements composing the phloem. We did work a lot in the geometry of the phloem in the trunks of trees, but less in the geometry of the phloem in the different vein orders of leaves, and pretty much there's only a handful of works depicting the phloem in the reproductive tissues that lead to the fruits, right? So I'll focus in these three major crops that we study here in the experimental station: Atemoya, which is a hybrid of cherry moya, mango, and avocado. And there is this paradox. I said in the beginning that avocado was by far the most cultivated, but we have only diploids of avocado, even though there are so many varieties. In mango, we have both diploids and tetraploids, and with Atemoya, we have diploid, triploid, and tetraploids. And so this is let's say, paradox. We have a lot of avocados cultivated, but that many genotypes available for working with, even though, as I said, there are so many varieties. And so the perspective I wanted to highlight today is the study of the phloem, taking into account the source and the sink organs. We normally know that the source and sinks are wired, but we are not going to pay attention to the wiring mechanism, but only to how this tubing system operates in the sources and sinks in this species that I mentioned, with the purpose of modeling source to sink transport in Buti systems, and eventually knowing what is the influence of water scarcity in these systems. So I'll start to tell you about our works in Atemoya, and we have two different aspects. One of them is the anatomy of the phloem, and then I'll briefly mention our irrigation experiments. And as I said, we have diploids, triploids, and tetraploids, which are products of backcrossing the F1 generation with parental lines, which are hybrids. And so we started to measure the anatomical features that could be interesting. For example, the leaf mass area was actually scaling with the ploidy. And so the diploids had the lowest leaf mass area, then the triploids, and finally the tetraploids. And this was also matching the cell size of important cells, for example, stomata. The diploid guard cells of the stomata were the smallest, but also the most, the denser, I would say. So their density was the highest, followed by the triploids, and then the tetraploids. So basically, we know that the ploidy increases size of cells, but the question is if all the cells increase the size similarly. And our focus was on the sieve tube elements of the phloem, which are these two tubes that are being highlighted, connected by the sieve plates. We study the phloem of the different vein orders, starting from the major veins, analyzing their width or their radius, and then their length as well. So we started by the major veins, and our results were very clear, clearly showing that especially for the radius, the polyploids have larger, wider sieve tube elements in the major veins, but also in the secondary veins, the third order, and the minor veins. In the case of the xylem, the pediole was a bit different, but they did follow a similar trend. So the phloem was a large, the sieve tube elements of the phloem were larger in the polyploids. And then we focus on the pediole because this is the area where there is this wiring, right? And we quantified the density of sieve tube elements. And what we show is that there is, even though they are larger, they have less density, both triploids and tetraploids. So we are now quantifying the total number because we did a similar study for the xylem and realized that the total number of cross-sectional area was very important for the total conductance. In the case of the zinc, the cherry moya has solitary flowers, and so we quantified their density. The tetraploids had denser branches, so branches which were shorter but with more numerous, with more number of flowers. And then the flowers of polyploids were both larger and wider than the diploids. So they did follow this trend of larger organs, as we mentioned in the beginning. And then focusing on the pedicels, we found this nice correlation between the length and the width, where the diploids had the shortest elements, followed by the triploids and the tetraploids. So we wanted also to see how this could be observed in vivo, and we developed this technique injecting a dye tracer that could be monitored in real time. And so we, after injecting it, the dye will accumulate in the final area where they should accumulate, which are the carpels of the flower. The flower is very weird. It's not like a regular flower, but it's typical of the Annonaceae, which is the family of the Cherimoya. And with this technique and doing image analysis, we could quantify the velocity of accumulation in the sinks. Either we don't know if this dye is going through the phloem or the xylem, because this was difficult to evaluate, but what we know is that in vivo, the dye is accumulating at this velocity, which was larger in the triploids. So all this information serves to establish a model, so we can model the hydraulic resistance from the source organs to the sink organs, and this resistance was lower in the polyploids compared to the diploids. That means they had more ability to transport photo assimilates due to the larger geometry of that conduits, both in the different vein orders. In the pediole, there is a constraint, so there is not so much of difference between ploidies, but then the flow is actually allowed to the reproductive organs at the stage of flowering. So then we wanted to relate these anatomical features with irrigation experiments. We did an irrigation experiment in the greenhouse with 3 different irrigations, monitoring the water in the soil and using the 3 ploidies, analyzing the different gas exchange variables. Just briefly, what happened is that after 2 years of experiment, we realized that diploids with enough water for irrigation consumed more water from the soil than the polyploids. But also, when we measure the assimilation rates or stomatal conductance or evapotranspiration, the results were similar between ploidies, but the diploids had a lower water use efficiency. And this was interesting, but then when we apply the deficit of water in the soil, the diploids were actually the ones suffering the most, shattering their stomata and impairing assimilation compared to the other polyploids. So this is regarding Atemoya, and with mango, as I said before, we did a similar approach. We started by analyzing the anatomy, and again, there was this correlation between ploidy and stomatal size in mango, and this size of the guard cells was inversely correlated with their density, which is the typical trade-off that is shown by so many polyploids studied so far. Then we studied the individual sieve tube elements in the different vein orders, and so we showed that both the length and the radius of the sieve tube elements were larger in the tetraploids compared to the diploids, and this was also true for the connection between the tubes. And this is a cross-section of the pediole with electron microscopy, just to show you how to find the individual connections, and which is not easy to find, but also how we measure the individual connections, which are the pores connecting the individual sieve tube elements. And the quantification of these pores just yielded the idea that the inflorescences, because mango is composed of very tiny flowers grouped in inflorescences, are actually showing a lower conductivity compared to the source organs. And this was actually interesting to explore. And this is a typical mango inflorescence from multiple flowers, normally only 1 or 2 fruits set. So we decided to explore the sieve tube elements at the top of the inflorescence and at the bottom. And the results were actually showing again that the tetraploids had larger sieve tube elements, wider sieve tube elements, but also that they were smaller than the petiole of the leaves. That means during flowering, it looked like mango is not allowing the transport of photoassimilates towards the inflorescences. And this is just showing that the tetraploids have the larger elements of the phloem. And this is reflected in the, again, in the modeling of the hydraulic resistance, which was, again, lower for the tetraploids compared to the diploids. So then we wanted to go further and explore the individual flowers. And first of all, the flowers of tetraploids were larger than the diploids. And then we wanted to see, okay, at the pollination stage, is there any role of the sieve tube elements that could be measured? So we dissected the style from the ovary and evaluated the sieve tube elements of the phloem. Again, the style had surprisingly larger or wider sieve tube elements compared to the ovary, and the tetraploids were always larger in both tissues than the diploids. And this was interesting because we think that the phloem is actually modulating the transport of photoassimilates to sub-organs within organs. I mean, during the pollination, the style should be supplied with photoassimilates, and then it's the ovary for fruit growth, the one that needs to be supplied. Now, actually, the fruits are larger of tetraploids, and this is a typical question that I have in different presentations. So to evaluate their resistance or their performance under soil water deficit. So we had a few trees in the field, so we decided to cut directly the irrigation for 2 years in half of the trees, evaluate the biomass accumulation, and the biomass was obviously larger in the well irrigated trees. And then we also monitored with using capacitance detectors of volumetric water content in the soil. And this showed that the trees without irrigation were actually having much lower water content in the soil, especially in the most superficial layers of the soil. We measured the leaf water potential, which was typically higher for diploids, but there were not so many differences between both ploides during the hottest months of the year, which are from June to September. But then when we cut the irrigation, diploids were doing very strange things compared to tetraploids. So basically their water potential dropped some days and not others. In the case of stomatal conductance, diploids had always a higher stomatal conductance compared to the tetraploids. And when we cut the irrigation, they dropped significantly their stomatal conductance compared to the tetraploids. They dropped it at a higher rate. And so what we're doing now is installing more sensors. And so we have a new project, which is called the 5G Tactile. And we have an experimental station of 40 hectares, and in collaboration with other members like Nokia company, we are actually creating the digital twin of the orchard, so we can model what's going to happen in a virtual space with the different mango trees. And I'm going to finish with the avocado. I think I'll be in time. With avocado, we have only diploids, and so we, instead of adding the variable of ploidy, what we did was to study the anatomy in regard to development. And I'll explain that. Avocado is an evergreen tree, but even though it's evergreen, it actually changes from the period of flowering, where the tree is filled with flowers, but it has almost no leaves because the leaves from the previous year are actually dropped almost completely. And then there is a burst of leaves, and this is correlating with fruit growth. So the question here is there this wiring between the leaves and the flowers at the flowering time? And so first, we evaluated over a whole year the stomatal conductance. And what we depict from this graph is that obviously the midday was the highest stomatal conductance, but normally the levels of conductance were typically low and maintained from the morning to the afternoon. Then we measured again the individual safety elements at the different leaf baying orders. And again, the situation was very similar to other species that we studied before. Basically, there's an escalation of the length and the width and the radius of the safety elements from the minor veins to the petiole. And also we know now, because it was not known before, that they have simple plates and this scaling relationship. And so when we talk about the zinc organs, the flowers, we talk about development. And so between flower and fruit or harvesting, more or less 12 months elapse. And during this time, what happens is that there is a change from flower to fruit, and the fruit just increases in size. So when we started to measure the biomass of the fruits, we realized that obviously flowers had a length to diameter ratio lower than the other stages. But what was surprising is that after the 7 month, there's not a change in the length to width ratio. So basically, the fruits grow proportionally, which is, it makes sense, but it's more surprising that the diameter of the pedicel didn't change between the 7th month and the 12th one. So the question is, okay, what's going on with the safety of elements of the phloem? And again, the flower had the shortest and the thinnest safety of elements, but then after the 7th month, there was not a significant change in size of the safety of elements, which even though the fruit really changed in size. So then we focus, maybe the things that change are the connections between the tubes, the sieve plates, and we measured the individual pores. And again, the flowers had smaller pores than the rest, but then this is how the flowers look like. And then after the 7th month, there was not an increase in the size of the individual pores. But more surprising was the fact that when we quantified the total number of pores per connection, there was not a change between flowers month 7 and month 12. So it looks like this is a conserved character that is maintained. So now what we are doing is to quantify the total number of safety elements, because it's the remaining question. So the increase in size in the fruits should be correlated to more tubes, given that the tubes don't really change their size or their connections. So we model the resistance of the safety elements in the leaves. There is a drop of the resistance, obviously, from the 5th order veins to the petiole. But then in the reproductive elements, there is a huge increase in the resistance towards the flowers. And that means that there is not this transport happening between the leaves and the flowers during the flowering time. However, during the fructification, of course, the transport is allowed from the perspective of the geometry of the phloem. And the last one, we are evaluating the water needs of avocado using also sensors, different sensors. And this is part of our project for precision watering and development. So we don't have still many results, but what we are using is the 5G and 6G technology to report many sensors in real time, including the information from drones and unmanned carts in our experimental station. And with this, I just wanted to conclude. And this is even though the species we are using have different phylogenetic origins, they share some of the characteristics, like the scaling relationships in the phloem, but also they change in the geometry of the seed tubes and the number of plate connections between them. And the way forward, we are interested in exploring the anatomical and biochemical changes of the phloem related with water deficit. Then we also are interested in water deficit and reproductive development. And finally, to use and develop novel polyploids and their resistance to water shortage. And with this, I like to thank all people in the field working in the experimental station, which are really helpful with all the experiments, all the collaborators, and all the funding sources. The last one is just thank you all for attending the presentation. And if you feel that this is an interesting research that we can collaborate, feel free to join us, and we can talk about joining our forces. Thank you very much.
Thank you, Juan. Congratulations for your work. Really impressive.
Thank you.
I'll save my comments for later, and I will pass the floor to Omar Garcia.
Thanks, Alexandra.
And Omar will talk about irrigation scheduling on banana crops. Can you share your screen?
Yes.
Fine.
And you see my presentation? Hello?
Yes, I
Okay.
can see it.
Okay. Well, thanks, Alexandra and Brunella, for the invitation. And oh, sorry. As I was commenting earlier to Alexandra and to Juan, my idea is not to show some results, but rather to take advantage of the FruitCREW members and trying to just establish some brainstorming or some discussion about the irrigation scheduling on banana crops. My name is Omar Garcia Tejera, and I work in the Canary Islands in Tenerife at the University of La Laguna. And here's one of the typical orchards that you can find there. So as you can see, it's a terrace, and they are very close to the sea and small holders mostly. And when I was preparing the presentation, I realized that instead of titling irrigation scheduling on banana crops, I should title, it's not changing. Okay. Towards irrigation scheduling on banana crops. As I said, the idea here is to trying to present you what have been done in banana in the past years, and just to gather some ideas from you guys on the new ways of improvement of irrigation scheduling on banana crops. So I'm going to keep with the presentation, and I would like you to connect to this WooClap questionnaire, because the idea is to have a more interactive presentation where in the next slides, I will ask you some question, and I would like you to participate and to give some comments. So if you can just go and register there, that will be great. I give you 1 or 2 minutes to copy the link. And just let me know when you are ready, so I can continue with the presentation. Okay. So just to give you some just view, a brief view of the crop. This banana is produced mostly in the tropical and subtropical areas of the world, and India is the largest producer with 34.5 million tons, followed by China with 11.8 million tons. And this map is kind of tricky because in Spain, in the mainland, we don't produce banana. Bananas are produced in the Canary Islands, and the same is true for France. They produce the the crop in the Martinique and Guadeloupe in the Caribbean and also in Portugal. But the islands are so tiny that they don't appear here, so they include it in the mainland part. Some facts or some things that you might probably don't know about the crop. It's banana are amongst the top 10 crops in terms of area of cultivation, yield and calories, and it's the world's most exported fresh fruit in terms of volume and value. So it's a pretty big crop. Also, banana are essential to maintaining food and nutritional security among 400 million people. And it's basically a staple crop, and it's like the basic for nutritional safe for many people in developing countries. Also, water is probably the most limiting non-biological factor affecting banana production, and the annual water requirements can go from 12,000 to 22,000 millimeters, depending if we are in the humid tropics or in the dry tropics. So we have a pretty important crop, both in terms of safety, nutritional security, and also in terms of economic revenue. It has a lot of water requirements. There are a lot of places where you have to apply irrigation in the banana, but this is not linked with research. So if you put the keywords banana and irrigation in Web of Science, you will have the result. It's 710 articles. Which is quite low if we compare that with the research on irrigation on olives. So here's the first question, and it's, are you familiar with the crop? Just feel free to answer. Hopefully someone got the link and we could, yes. Cool. Yes, nice. So we have, I count 20 people. Here we have 8. I don't know if all the people just managed to, no, 16 people. Okay, half of the people, more or less, have answered. So most of you know the crop. You have seen a banana tree. So I'm going to pass very fast through this slide. So basically, this is a representation of a banana where we have the main plant. This is called the mother. As you know, banana, it's a rhizome. It's like a big potato. I explained that in that way to my students. And what is interesting is that the plant has not, it doesn't have a real stem. It's called a pseudo stem because it's not composed of wood, but it's made of folded leaves. So this is not a real stem. It's just the leaves folded, just create this pseudo stem. Then we have the fruit, which can weight around 30 kilos if the yield is good. And from the irrigation perspective, few facts that you might find interesting is that the leaves are quite big. So the area of the single leaves can reach 2 to 3 square meters. The leaf area index is around 2 to 6 meters, square meter of leaves per square meter of soil. The root system is quite shallow. It's around 0.5. It's really it's hard to find the rooting depth below this depth, this 0.5. Also important is a latissiferous plant, which means that if you cut the leaves, the plant start to exudate latex. And then another interesting thing is that the plant presents diurnal change in leaf angles that do not respond to variation in soil water content. So you might have the water content and field capacity and still having this trend in the leaves. So basically, when I'm talking about change in leaf angle, is that this, I don't know if you can see my pointer. Hopefully, yes. So basically, this part of the leaves just fold, and it folds completely. So the leaves point to the ground, and then it opens. So it's like a kind of an umbrella, or like the wings of a bird, if you want to see it in that way. And here you can see some of the recorded changes. So here's during the day when the vapor pressure deficit is high, leaves tend to fold, and then as the night comes and the BPD is lower, the leaves start to unfold. And this has a lot of implication for the radiation interception and the transpiration of the plant. So what about the banana crop in Tenerife? This is the island, it's Tenerife. The island has an active volcano here called Teide, and Teide has a height of 3,700 meters. So the island is very steepy from the sea to the top of the mountain. The cultivation is mainly in this northern part of the island and also in the southwest. And the weather here, it's very, we have very sharp change in the weather. So the northern part is more humid. It's usually covered by clouds, whereas the southern part, it's more like a desert. In fact, here in this part, the precipitation barely reach 100 millimeters. The water prices for banana irrigation are quite high and can go from 0.5 to 0.9 euros cubic meter. And the sources for water depend on the area of cultivation. But basically, we use galleries, desalination, and reclaim it in different proportion, depending on the area. And the average irrigation applied to the crop, it's quite high. It's from 8,000 to 11,000 cubic meters per hectare. So this is more or less the characteristic of the banana cultivation. And also the main band of cultivation goes from 0 to 300 meters above sea level. So it's quite close to the sea. This is a zoom of one of the areas. This is called Isla Baja, and you can see that all these green parcels are banana. You have like open field and also in greenhouses. Just a funny fact that some municipalities in the island has forbidden the use of greenhouses because banana is part of the landscape, and Tenerife is a very touristic island, so they don't want to have greenhouses in their plantation because tourists want to see the banana like in the open field. Also, this is an ancient volcano here, and basically it's used now as a water reservoir where we pump the desalination water and the water coming from galleries up to the volcano, and then we use that to irrigate the crop. So how the banana cycle works? Basically, you start with an initial planting. This plant grows and you have different suckers. And by the time that the plant flowers, you have to leave one sucker that will become the plant or the crop in the next season. So basically, when you harvest, you cut this initial plant and you leave the sucker that you decided to leave for the next season. So in the next season, you start the cycle again. You have here the mother, here the previous sucker, and then you have different suckers. And then when the flower appear, you have to leave one sucker. And at the end of the second cycle, you have here the grandmother, here the mother, and this is the son for the next season. And the cycle continues on and on for 5 to 8 years. And at that point, you or farmers usually restart the cycle because the planting lines just become not that straight. So it's difficult to manage the crop. And because of that, because you always have a green area, the crop coefficients remain quite stable. So you have an initial trend up at the very early stage when you start to plant it, but then the Kc fluctuates from 1.1 to 0.7 more or less. And also the evaporation from the soil is quite low because farmers tend to leave the residues on top of the soil. So evaporation, it's basically hindered. So here is a question for the FruitCREW group. Which plant-based sensor would you use in this crop to schedule irrigation? And I'm going to open the question. No one wants to participate here. Yes, drones. I don't know. Thank God. Cool. Subflow. Zim. Nice. Cool. So for those of you that have proposed a method, just good news, at least in my opinion, is that little work has been done. So if we add, for instance, subflow to the keywords, only 2 paper just pop up compared to the 191 that you can find if you change the word banana by olive. Also, there is no specific literature for dendrometer, not even for trunk shrinkage. So there is no, at least as far as I in my research, in my browsing, I didn't find any literature related with this type of sensors. Only one research related to lift turbal pressure use, and remote sensing was mostly used to characterize farms in terms of heterogeneity, and also to basically to monitor some diseases, Fusarium oxysporum, which is the main disease that affect the crop. So here's another question: how would you measure leaf water potential in this crop, given the fact that leaves are super big? Interesting thermal image. SPC, I don't know what it is, but maybe during the comments we can just talk about. So one of the, if you remember from the previous slides, one of the key things that self apart banana from other crop is the latex. So the problem is that if you use a regular Scholander chamber to measure leaf water potential, it's very, very hard to distinguish between water and latex. So these guys here, Milburn and et al., just came up with a solution, which basically what they do is they measure the rate of latex exudation, and they use that as a proxy for water potential. So basically the idea is when you cut a leaf, the latex start to go, the exudation rate is very high, and then it goes down, 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 down until the water potential, the pressure water potential inside and outside the leaf, it's balanced. So at that point, theoretically, if you measure osmotic water potential, you can use that as a surrogate for the leaf water potential. And that's what they do. They basically measure the latex, the osmotic potential of the latex when the rate of exudation it's low. So what is the ongoing research? And that's why I wanted to just engage you in this presentation and to collect some ideas. The first one is to test dendrometers to manage irrigation based on pseudo-stem fluctuations. My hypothesis is that it's going to work up to a certain point because when the fruit just appears, the demanding, it's going to be like a very, very, very strong sink, and the signal, the trunk shrinkage, it's going to be hindered. So it will give you some nice signals of or nice variation up to this point. But this is an hypothesis. We have to test that. Another one, it's or the second point, it's installing some subflow sensor based on compensated heat pulse. Some of you just name it subflow sensor as a possible plant-based sensor, and I agree that it can have a lot of potentialities. And in fact, some previous researcher have used the subflow, the Granier method, to monitor the transpiration of the plant. Basically, they first did this nice experiment where they dyed the water and they just stained the plant and observed that the main conducting part is the center cylinder, this part here, which corresponds with this part here in the plant. And they decided to install probes in this part, in the center cylinder, just to corroborate the dye experiments. They decided also to install probes not only in the central cylinder but also in the cortex. And basically, this is what you are seeing in the graph on your left. Here you have the trend of the transpiration of subflux density in the central cylinder, and the lines represent transpiration or subflux density in the cortex. So as you can see that in the center cylinder, you have this nice bell shape of transpiration. Also, they did some analysis on comparing potted plants, the daily subflow of the potted plants with daily gravimetric water loss. So basically, they compare the results from a microlithimeter using pots with the daily subflow, and they found this very nice correlation. My point here is that banana has a lot of water. It's really, as I said, the pseudostem, it's mainly composed of fresh tissue. And also the water content of the plant fluctuates a lot throughout the day. In fact, the authors found a 42% variation in water content in field plants. So my question is that my concern is that if this variation is going to affect somehow the determination of the subflow using any of the subflow techniques, because I know that the Granier method has some problems when there are changes in the heat diffusion through changes in the water content. So here's another question. Maybe if you have experience with subflow sensors, have you ever used subflow system in plants with significant fluctuations in internal water content? Ah, yes, nice. Yeah, I would like to hear the person that says yes. I would like to hear his or her comment later on. And what about modeling? Just one of the parts that it's inside the FruitCREWS is the use of the developing of DSS and use some modeling to improve irrigation scheduling. And I found this nice article that can be a good lesson for us. Those researchers, basically, they they did this nice modeling approach of water flux to improve banana irrigation scheduling and management in an area of Colombia. They apply a very easy but robust approach for the modeling of irrigation, where basically they model the water balance using crop coefficients and effective precipitation. And they found that the farmers recognize the advantage of the models in terms of reducing water, but at the same time, farmers were reluctant to change long-established traditional irrigation management practice. And why was that? Because most of the farmer in that area do not understand concept like soil water deficit, the impact that ETO has on the crop water use. And also that's very interesting. They work in based on time rather than depth. So they developed this nice approach where the recommendation were based on depth and millimeters of water, and farmers wanted time to schedule their irrigation. So here is my last question, and it's if you have experienced the same constraint when trying to integrate novel tools in your irrigation area. Yes, so it's a common trend. Nice. So my point here is that just after thinking on the inclusion of modeling in our ongoing research in banana, and maybe that's a lesson that should we consider for the FruitCREW for some of the board packages that are in the FruitCREW group, is that perhaps it's preferable to opt for a simple model that enjoys broad acceptance rather than a more precise and mechanistic approach that lacks widespread approval. And from this simple model, just start to do some iteration and just to teach farmers on how to use and starting to incorporate more complex elements. And thanks a lot for your time and for being here and for your participation in the surveys.
Thank you very much, Omar. Very interesting presentation. So I will interrupt the registration.